What is one thing that cholera, TB, many historical plagues, staph, and the common cold have in common? Of course, they are all devastating to different degrees, but they are also all caused by bacteria. So how can bacteria, these microorganisms that we know cause diseases and suffering, ironically house the treatment for diseases like various cancers and lung diseases? To find the answer, we need to look at a war spanning the last 100 million years. This is the war between bacteria and viruses. Bacteria-specific long-standing viral enemy are bacteriophages. The spidery-looking bacteriophage latches onto a bacterium cell membrane, injecting its venomous RNA or DNA into the bacteria. The bacteriophage is ready to take over and even kill the bacteria. At this point, it looks like the bacteriophage is winning this war. It's ready to replicate itself and create an unstoppable army of robotic bacteria-killing clones after taking over the bacteria's machinery. However, bacteria are not helpless. For bacteria to survive for all these hundreds of millions of years, they had to find a way to fight strategically. This involves a part of the bacteria's immune system embedded in their DNA called the CRISPR-Cas genes. In bacteria, CRISPR refers to the Cas and CRISPR genes. When the virus genes enter, the CRISPR genes create RNA and the Cas genes create proteins. Cas proteins have enzymes, carry CRISPR RNA, and race to the front lines to destroy the viral genetic material that invaded the cell. CRISPR RNA directs the blind Cas protein vehicle to find a spot on the viral genes to bind to on its complementary strand, like two puzzle pieces clicking together perfectly. Finally, the Cas protein makes a cut and the viral genes are not effective any longer. Now, how is this all translated to actual medicine that could be used on mice and even humans eventually? Recently, two scientists won the 2020 Nobel Prize in Chemistry for discovering that CRISPR-Cas9 could be used for gene editing to treat various diseases. As a disease treatment, the large CRISPR-Cas protein vehicle carries CRISPR RNA and a guide RNA and another DNA/RNA strand. Like the CRISPR finding a complement in viral genes, the CRISPR can also find its complement pair in mice and human genes, and even delete or add a new strand. There is another CRISPR RNA that we need to talk about, called CRISPR-Cas13. So this pair, instead of DNA, this remarkable pair binds and cuts RNA. Have I mentioned how much bacteria despise viruses? Well, CRISPR-Cas13 has been a COVID testing tool to identify and cut up COVID viruses. Scientists are starting to find uses for Cas proteins 9, 12, 13, and so forth. They're also adding additional enzymes to attach to the Cas protein vehicle. We're only at the beginning of a revolutionary event in biology, and I can't wait to be a small part of that. Thank you so much for watching.